Welcome to this episode of Straight Talks Unedited. You know that this program we do very frequently with the range of topics. And now the topics is nothing but the COVID. And we have been talking about it for a while. We have been talking to doctors. We have been talking to public health experts. But we need to talk to the people who, end of the day, find out which is what, which variants are coming, how the genes are changing. So how the game of gene is being changing. So I have the best person in India right now with me to talk about it. I have Dr. Saumitra Das, Professor Dr. Saumitra Das. Dr. Das, welcome to the show. He's the director of National Institute of Biochemical Genetics in Kolani. He's a professor in Indian Institute of Science. And most importantly, also the main coordinator, the head of INSACOG, the nodal body, the umbrella body that have so many institutions. It started with around 40, but I think now the numbers has increased a lot, which are studying the genomics of this COVID virus, which is extremely important to understand the way public health and the society at large should respond to this pandemic, which is being raging throughout the world. Again, welcome, Dr. Das, to the show. I think it's really a pleasure and privilege to have you on the show. Let me start with a very direct question. This Omicron is apparently raging through the world. So let's talk, because you've been studying this. How is the situation in India right now on Omicron?
See, the situation is actually in the metro cities. I think Omicron is spreading like, it's just some of the metro cities, it has already surpassed the Delta. And in the other cities are also, it's picking up. We are recently actually had a scaled up sequencing in the metro cities just to get an idea that how fast it is actually spreading in the metro cities because of the international traveler and the exposures.
Right.
And the results clearly suggest that these metro cities are actually very high on the Omicron. But, in a span of maybe another few weeks, maybe it will just spread and ultimately replace Delta in most of the places.
Right. So you were saying that the numbers are very high in metro cities. Just yesterday, we had the figure of West Bengal or Kolkata is about 70% plus in Kolkata. I think in Delhi and Mumbai, they're also very high. But overall in the country, we find the numbers rising with more than 1 lakh every day new COVID patients. And a new study has suggested the number may actually reach to 50 lakh by January 8 and February. Your thought on that? Will it be going to be so large, Dr. Das?
So this is very difficult to anticipate, because I think these prediction models are not always correct, but they are useful. And one way to think about it, because it is mild and most of the time asymptomatic, the patients who are actually carrying the virus are unknowingly spreading the virus to their family members, relatives, and associates.
Right.
So, and that's the main reason. If you really compare with the wave 1 and wave 2, with the original your Wuhan strain, and then followed by the Delta in the second wave, this is a major problem that the isolation is taking a little longer, because there is a window of between the first manifestation and the detection. And in that window, they are already spreading to their associates. So that's becoming a reason.
Okay. So what is the figure in metro cities generally? Is it more than 70% or 80%?
Right.
How
In some of the metro cities, it is actually more than 70 to 80%. In some of the is actually near about 70%. So it all depends.
May we know which are the most worst
pockets
affected so far? May we know which are the most worst affected so far? Which are the metro cities?
I mean, as you also see in the media, it's mostly affected in Maharashtra and Delhi. And now you are actually also seeing it in Kolkata. It's picking up. But as and when I think the results are coming out from different places, we can see a change between December and January. And January, most of these are actually, surpassed by the Omicron. The Delta is replaced by Omicron.
Okay. Also, coming to West Bengal right now, you said before that West Bengal is very quickly shifted from about 20%, 15 to 20% in the December to about 70% now, as the official figures are suggesting. So, is it mean the end of Delta? What's your thing?
Well, end of delta means suddenly, yes. What happens actually, the virus replaces one strain. You have seen that delta has replaced, and before delta, it was D614G. It started with Wuhan, and then there was a mutation which actually,
Right.
incidentally, we reported from in Calcutta also. There was 9 sequences we did from Calcutta population, and we showed that D614G in this population.
Oh, so it
That was
creates.
in 2020,
Please
2021, early 2021.
please carry on.
Right. So this D614G actually somehow outplayed all the other strains, all the others clades. But then Delta came, Delta replaced all of them. Actually, Delta also have the D614G. And now Omicron will do it. So in future, we really don't know whether the Delta will come back. It doesn't really look like. But what happens, there will be mutations accumulated in this particular background. It's already, we are seeing there are some sublineages coming up from Omicron, which we call BA.1, BA.2, BA.3. They are something like a grandchildren of Omicron started coming out.
So very important. So you say the sublineages are already coming out from Omicron. Now, the point is, are these sublineages, as per the whatever analysis you are doing right now, are they in terms of infectivity and virulence same as the grandparent, or is different?
See, these things actually needs to be experimentally verified.
Right.
And what we need to do is essentially to establish a clinical correlation. As you know that so far in most of the cases, Omicron is not so severe,
Right.
right? And the other sublineages are very less and rare in numbers. And so we don't have enough data to predict whether it will be more or less severe. Uh, but they are actually coming up, and then the whole purpose of this INSACOG to continue this surveillance to look for these sublineages whether they are
Right.
going to get a momentum and then come up with a huge way, or any
Okay.
other new variant is cropping up.
Very important new variant you just mentioned about the new variant. We have heard that in France, the new variant 1H IHU being kind of that being found out. Any information about that in India IHU so far?
So far, I am not aware, but this is, it's not a surprise that there will be new variants keep coming. Time and again, people have been saying it. This is RNA virus, and the mutations is very common, and the mutations that favors the virus will be always selected. But at the same time, there is pressure because of the vaccinated population. So virus is also forced to escape the vaccine challenge, and
Right.
that's how they also tend to mutate more faster. So, but as and when it comes, we have to see it. There is a new mutation, as you rightly pointed out, this is called IHU variant, which was first detected in southeastern France, I think, which has 46 mutations and 37 deletions in the nucleotide level. That results almost 30 amino acid changes and 12 amino acid deletions. And some of these mutations could be relevant in terms of the infectivity, because if there is a receptor binding domain in the spike protein which makes contact with the ACE2 receptor. Now, if there is a mutations that favors the affinity or interactions with the ACE2 receptors, that will be definitely increasing the infectivity of the virus. But again, that needs to be tested, and we need more data to see whether it's causing more severity or infectivity or transmissibility.
No, that's extremely important, and I hope that in near future we'll be having a outline of understanding that what ICU is actually doing or not doing in India. But let's go back to Omicron. In Kolkata, we find that Kolkata and Howrah have been the two most highly districts with highest positivity in the country right now. The data that we have from the government of India is from the week from 28th of December to 6th of January, was Kolkata with 53% positivity rate and Howrah with 39% positivity rate, are within the top three. Now, as you said, the Omicron is raging through the all metro cities. Why you think that positivity rate is so high in this part of the world, in this part of the country, rather than the other metro cities? In Kerala, it's around 12%. In Mumbai and Delhi, also it is reasonably low. So how it can be explained?
No, I mean, one is definitely the exposure
Okay.
of the to the virus. And now the exposure can happen from the international traveler. And if you see it in the beginning, most of these Omicron cases were actually restricted to the
Right.
international traveler. But slowly, it becomes a contacts, and from the contacts, we started seeing in the community.
So now it's the community transmission. Now it's a
Now it
community
becomes a
transmission.
community transmission.
Right. Right.
So, you have to somehow correlate the exposure to the foreign travelers and how good they were restricted or how fast they were actually quarantined probably will have an impact on the spread of this particular virus in others.
Okay. Uh, One thing, Dr. Dash, is that doctors are saying that though the infectivity is very high, the severity is less. Even clinically, they are finding it in comparison to Delta, the severity is much, much less. Can it be explained from the genetic point of view? Are you also finding that any gene or whatever or sequence responsible for the severity is not there, perhaps in Omicron? Can it be explained from that end?
No, so there are two angles. See, forget about only Omicron. Any strains, if it is the alpha, beta, gamma, delta, the severity depends on the individuals
Right.
because of their inherent immunity and also their prior exposure,
Okay.
their vaccinated status, their comorbid situation. So
Right.
all these factors in to the severity in a particular individual. So we have seen the same virus may not cause the same kind of an impact in the same locality and also in the same family because of the individual get-up, as you rightly said. There could be genetic determinant, there could be microenvironment. They all actually contribute to the disease severity.
Uh, let's go to the INSACOG, because INSACOG, how many institutions right now you have under your belt, Dr. Dash, in INSACOG?
So it actually started with 10 institutes, and
Right.
this is first time Government of India actually asked us to launch this program, and it was across CSIR, DBT, DST, Ministry of Health, ICMR,
Right.
all of them, and we had representative institutes in different regions covering, and 10 which we now call a hub lab started this exercise sometimes in December in 2020. And then we started probing, and that's the time when we started looking for the UK variant, if you remember.
Yes,
which we
yes,
now
yes,
call,
absolutely.
Right. So this alpha variant were actually very scary, and we started looking for it, and that's the time we came out. And then we slowly, the Delta came, and
That was the first wave. That was the first wave.
It was during the end of the first wave.
Yes, toward the end of the first wave.
Right. And then we actually figured that the numbers are rising so high and, it's just spreading in all over India. We need to reach out to all the districts so that we have the proper samples in hand to make a comment on it. So we extended to some satellite centers. They were actually connected with the hub labs. And then we increased this to another 18 state laboratories, which are also, somehow mentored and then trained by the hub lab to sequence and analyze and help the hub lab. And eventually now we spread it into, I think, a week back, it was 43, where we have this INSACOG network had spread it. So the centers are all over India, and some of them are new, but we are just encouraging them, keeping in mind that in case these numbers becomes unmanageable, then I think they will be coming really handy. And also this is, as I told you, this is first time, this is across the agencies, this consortium is formed, and then it's actually functioning well. So in future, this kind of network can be used for any future pandemic. That is something very important.
But don't you fear that any quality can be compromised with the numbers being risen to such a...
Right. So, we tried to have a control on that. So what we did is essentially we have a technical advisory committee who analyze and assess the ability and the quality of the sequences done by the new inducted centers
Right.
and even the existing centers, and try to see their whatever predictions or whatever analysis they are doing is actually correct. So that we ensure the quality control. And we usually meet on Wednesday, where we all, the PI and co-PI of all these institutes, I
Right.
meet with them along with Dr. Sujit Singh, who is the co-coordinator of this INSACOG, and we just try to analyze what is their issues. And we also meet on Friday, where we have a scientific and clinical advisory group, where
So they also look after.
convener, and then they analyze the problems, and then the policy decisions are taken and then recommended to the appropriate authority.
So the quality control is assured, whatever the numbers being increased.
Definitely, yes.
That's
And they
part
are
is taken
also,
care of.
actually, I told you that the newly inducted laboratories are under mentorship with somebody else. So somebody actually mentored them, make sure that they're doing well, they're not having any issues.
Okay. In this context, I just would like to take few questions. I spoke to few public health experts and doctors before having this interview, because I wanted to have their opinion. They had some questions to ask you. They may not have the platform to do that. So just without taking anybody's name, I'd like to take few questions which I have with me. Number one is that every day in paper, in Government of India's publication, we know what is the latest number of COVID. What are the active patients? What are the every figures are there. So, is it possible? Some public health experts are kind of asking to for the INSACOG to come out with a regular release of percentage of variants in samples across India, so that as doctors they know what's coming, what's incoming. Because some doctor is saying, say when I'm treating a patient today, uh, uh, the INSADOC has said that in Kolkata 70% is Omicron driven as of now. It means that around 20-25% should be Delta. So when I'm handling a patient, I really do not know that what exactly is coming up. So I have to go by the clinical symptoms. So if some weekly, regular kind of official output be put up that this is the situation in this metro city, Mumbai, Kolkata, Delhi, Bangalore, it's easier for the physicians to respond to that. Your thought on
I'm
that?
a little surprised with your question, because we are already releasing a bulletin every week for
Okay.
last couple of months. And this weekly bulletin is put up in the web page of Department of Biotechnology, where we have a link to INSACOG. And the weekly bulletin, exactly this is the purpose. We try to aware what is actually happening in the country and what is the way we are moving, what is the sampling strategy, what is the global scenario, and what we intend to do. So maybe
But do you go beyond national level here? Are we going to get data of the regional data, or is on national?
no. So see, there are two angles to it. One is definitely we have a much deeper report which goes to the government, and
Right.
we have something which we actually share with the public domain. So that, as you say, that it's definitely, I appreciate the concern of the clinicians, and we try to cater that, and we try to keep them informed as much as we can in terms of this emergence of the variants. And also there we
Right.
mention about the global scenario and how India is doing at
Right.
par with the global scenario.
Okay. I just wish that this data, because the data sometimes get lost among the maze of data everywhere. So if this data can be made much more directly accessible,
It is
then it
accessible.
might
It
be
is
easier
accessible.
for the
Anybody can access it. If you just have to click it into the Department of Biotechnology,
okay.
DBT, and if you will see in the front page, there will have an INSACOG highlighting in link, and there you can see all the bulletins.
Okay, so it has every week you are. So I think for the physicians and the public health experts who raised this question, and for everybody else, Dr. Das very pointedly said that every week they are coming up with the latest kind of situation, and that's a region-based situation. So somebody in Kolkata or Mumbai or Delhi know that what exactly is happening there. So you just have to go to the Department of Biotechnology and click to the INSACOG link.
INSACOG web portal. And
INSACOG portal.
in addition to that, we also have two portals. One is the INSACOG portal at IGIB, is another
Okay.
institute, and another INSACOG portal at NIBMG, where also we have an update, and you'll have more scientific information. Because see,
Right.
the bulletin is more public consumption, but more
Okay,
scientific analysis will be actually available on those portals.
great. I think very clearly responded to that. Another question is that the sample size. When INSACOG started, we heard that around 5% samples you tend to look at. Now, we clearly understand the enormity of the situation. I think nobody has thought at that point of time that we'll be handling so millions of cases every week or every month. Right now, India is having about 1 lakh plus every week. So every day. So what's the number of cases you are handling right now as an INSACOG in overall and also in West Bengal?
See, what we believe that it is not the numbers, it's the sampling strategy.
Yes,
And you have to make sure that you have a representative sample sequenced. The percentage
right.
really doesn't help
Everywhere.
much. Right? You can easily have a proper sampling and figure it out. So what we consider, there will be a basal level of sentinel surveillance, which we try to get it from every district all the way, irrespective of the current situation or the previous situation. But in addition to that, we try to have all the vaccinated people earlier, at least all the vaccinated reinfected people, we just try to sequence there. Then we try to have a surge districts or surge events. Or suddenly you can see in one particular area, there are certain, some colleges or some universities are suddenly reporting there are so many people are infected.
Sudden
Immediately
spark.
we try to get the, yes, as a spark, which we call a surge sampling. So that also we do it. And in
Right.
addition, in case there are some report on severity of a particular districts or a particular mohalla is reporting that their people are getting more sick compared to others, immediately we try to sample that. So that is a dynamic situation. We keep, as we said, that every week we meet and we discuss that whether we need to revise our sampling strategy. Then we have NCDC with us, National Center for Disease Control, who actually are experts of the epidemiologists also participate in that, and they take a confirmed decision that we need to revise this and then relook at this. Just mere percentage won't help, because
Won't help.
that we might miss. And another thing that I am trying to alert everybody, that while we are only going after Omicron and Delta, we should not consider goal with a counter attack. That there could be another variant slowly
Right.
cropping up, and that is more important. What known enemies you already know, but the unknown enemies is something I think the surveillance should continue to make sure that we can detect them early and see that something else is not cropping up from our country
Any
or our.
unknown variant in the horizon right now?
There are so many grandchildren of the deltas. This AY series has gone up to so many numbers. So we keep on seeing it whether they are crossing the threshold of 5% or not. Most of the time they are actually less than 5% or even lesser than that. But we keep reporting them, try to make sure, and then we try to docket them and try to see with the evolution of the variants. And we have a data analysis team where NIBMG also helps in that heavily. And we try to see an IGIB along with them. We have ILS Bhubaneswar along with us. We have CDFD along with us. We have a big group for statistical analysis and also this computational and this variant analysis. So we are actually pretty much aware that how these things comes. And we have seen it earlier that between the two waves, you can see the diversity of the
Right.
variance decreases. Suddenly you can see, and that is a signal that one more wave is coming. But when you have a situation, we can see that suddenly one strain, Omicron or Delta, is coming up, and the others are going down. So then you know that this another wave is coming, right? So those are the evolution things that we keep
So
doing
it
it.
means that a new variant, for the layman's understanding, a new variant normally triggers a new wave. Can we
Exactly.
put it that way?
Exactly. Right. Right.
Right. So another question that has come up, that so far, as per the government of India's data, the number of Omicron cases are being peaked at 3,600 something, as per the latest data. Now, you have just mentioned that most of the metro cities are about 70% cases, and I know that in Kolkata, your figure is based on about a sample of about 500. So, just a simple calculation: out of 500 samples, if 70% is positive, it means about 350 being positive. So that's, I'm talking about Kolkata. But we see the official figure of West Bengal around 30, around 30. So how it can be explained? So there is a separate set of protocols. How you kind of officially recognize something as an Omicron, or is not part of the sampling process? Just if you can throw some light on that.
So, we have, as I told you, there is a strategy, and at some point we were trying to have more sequences done between 21st to 31st of December, and in a metro city, which we call an escalated or accelerated sequencing, or enhanced
Right.
sequencing in those pockets, try to see the situation. Now that we know that 70 to 80% is Omicron, we need to revisit and see that other than Omicron, whether something else is also coming up.
Right.
So we are actually now more interested to spread into districts and even into the rural, remote places, and try
Right.
to see whether we can sample some from there and try to see whether we can figure out that something else we are missing or not. So as I told you earlier also, it is very important that already you know that this has come to 70 to 80%,
Right.
but it is now more important to look for something which is unknown.
Yes, that's extremely important. The unknown enemy around the corner is very important to know. Just to quickly go before going to the next topic is the numbers you were talking. The total numbers of Omicron right now. Sorry, not Omicron, but the samplings, genomic analysis right now in India. How we should put it? What is the figure generally? I am talking about a 7-day average or something. How it can be in India
No,
and
no,
West Bengal.
this is again a dynamic situation. You have to remember, see, there
Right
are,
now, you are
when
talking
we
about
take
the
it
present situation.
decisions, see, the numbers I won't remember, that's the bulletin
Not
will
that
show
important.
you. Bulletin will show you. But what I will tell you that there are some human factors are there,
Right.
because sometimes it's a transport problem. So the remote places, they takes time to transport the samples. It's sample collection is one thing, but that has to reach to the sequencing centers. So there is a challenge there. Second thing is the people who are sequencing, they are also getting infected.
Right.
See,
Right.
and this time I can see the hit even in our NIBMG, because the spread is so fast, and I can see, we are really struggling to keep on the show. Although I now diverted other manpowers to continue the show, but some of the centers may not have that luxury to get additional people
Your
to
Kolari
run.
center is having the entire West Bengal and Northeast, I believe.
Yeah, we are
It's
catering
such a
that, but
huge
it's a
command
huge
area.
place. And not only that, see, others are also having problem in other
Right.
centers. There also people are getting sick. Third thing is that sometimes, not always, you have an availability of reagents is a problem, because sometimes, because of some reasons, Christmas time and other reasons, it gets stuck. So, what we talk all the time in the media is the number of sequences done in India is less. But what we don't factor, that number of
And it's being
sequences
compared with the
took
developed
place.
countries. It's being compared with the developed countries.
That's one thing. And second thing is essentially we are actually catering a huge population and in hugely remote places also.
Right.
But the sampling, we are actually trying to do it intelligently so that at least we don't mess it. Even if we are less in number, but if we can actually correct way of sequence and correct way of sampling, then I think we can beat the numbers.
So what basically you are saying, Dr. Dash, is the quality of the sample is very important rather than the quantity part of it.
That's
So you are
basically it.
ensuring the stratified sampling. You are ensuring that the samples are representative. So it's not very important to know that percentage is 5% or 10% or
No,
1%.
no,
It's more
no, no.
important to ensure that it's representative and includes all over the country. That's what exactly you are driving at.
Well, although I also appreciate that the numbers should increase, but we have to keep it in mind, and you have to balance with the machine power and the actual manpower.
Any plan in the, because this COVID is now staying and emerging and all that, any plan to kind of enhance the number of sampling in the near future?
Right. See, what we are actually trying to do it, we are definitely keep on having increasing the centers so that we'll have more and more people involved into this network, and we can extend it. In addition, what I was trying to see, it's not the increasing in numbers. What is important that once this, let's say this third wave
Right.
goes and it gets into an endemic situation, we are actually interested to start sewage surveillance. The sewage surveillance is something which if it goes in the back, it can indicate prior to the actual onset of the variant. So because, in the polio network also people have done it in
Right.
earlier. We are trying to take help from that polio network and then trying to do it in a similar way, sewage surveillance in different cities. We already started doing sampling. So essentially we are trying to see, can we really detect the emergence of the variant much earlier than the clinical onsets. So that is one thing. Second thing, it is very important to somehow connect the clinical network. As you
Right.
said rightly, the clinicians are sometimes in dark. They have no clue what's actually happening. We are now consciously trying to involve them and trying to have a clinical network along with this INSACOG so that we know that the patients who are hospitalized, mild, moderate, severe, we can actually connect their status with the emergence of the variant.
Okay.
So
So
then we'll be able to
this
accurately
network has
tell.
started. This
This,
network.
this already started pilot scale, but we are going to expand. That's basically the future expansions are in these directions. Not only
Okay.
the increased numbers, but we are trying to increase our horizon by including some more centers where we are less represented so that we don't have any struggle to get their samples sequenced
Okay.
in the remote places, sewage surveillance, and the hospital network.
Okay, we have almost come around 5 minutes time we have. Just very quickly, as you said that you would like to expand the horizon on sampling. There's one thought about the universities. I think the universities around the country are, many of them are equipped with a lot of manpower, quality manpower. Any thought about including the universities or the institutes, scientific institutes, which are still not there in the process? Is it
Right, right,
possible?
right. So we are always open. So there is a procedure in place we
Right.
call an expression of interest. So first we get an applications from, let's say, one place who have started sequencing. Then we immediately try to connect them with our, as I said, the technical advisory committee. They try to see what kind of machine they have. Do they have a nanocore, or they have an Illumina system, or they have a iron torrent? So try to see their capacity of the sequencing, whether they have some experienced people available, whether they will be able to support their manpower, what is their ability to turn around time. All these things we evaluate. Then we discuss it in the meeting, and then we
Then you.
communicate with them. So that
Okay.
is also, so everybody is open and definitely allowed to apply for
Right.
getting into the INSACOG.
So definitely you are expanding the horizon on that line. Uh, Before I kind of wind up with future, uh, a quick question that has come up from one of the experts is that we know this citizen Omicron has come up from a particular country, originated in a particular country, should not kind of name any particular country, but Delta is also from this part of the world. So we know the origin. Now the Omicron is talked about from the pig, and the Wuhan virus from the perhaps pangolin. So but we have never heard about the origin of Delta. Any pointer on that? That from where Delta came?
No, no. So these are all assumptions
Uh-huh.
because, as I told you, that in any RNA virus, this is very common, and RNA virus itself does not have any control on how they will mutate and where they mutate, right? So we won't know, and if you really see, Delta mutation was not too different from the other mutations.
Right.
Omicron is something like which was really, came in as a surprise because there are 30 odd mutations in one particular protein. But see what happens in general, the virus try to escape any challenges. So whenever there is a vaccine or some kind of a challenges they face, they will try to somehow mutate and bypass that. And,
Mhm.
and, and that could be a quite natural thing. So I won't really see that how it happens. But
Okay.
how it's spreaded is something, that is our power. So we can actually make sure that we can detect them early and contain them as much as we
Okay.
can,
Okay.
restrict the spread.
Okay. Another very quick question is that uh, the vaccinated patients right now seem to be having less severe infections than the others, even in case of Omicron cases. So, any genetic linkage to that? The vaccinated patients are, you're finding the positives which are with vaccines. They are kind of have any genetic reasons that why they are more protected?
No, so the vaccinated people are expected to be protected better because the vaccine, I've been saying it, vaccine does not uh,
I have a
complete
supplementary
prevent.
question also, Dr. Dash, if you respond together. In Kolkata Corporation area, the figures that I have with me right now, it's showing that out of the total positive cases, about 70% or more are double vaccinated, are fully vaccinated. And in the hospitalizations, in the last couple of days back, out of 22 hospitalizations from KMC area, in a particular sample, 21 had double vaccinations. So does it also mean that Omicron, as it is leading the surge right now quite clearly, is evading the vaccines that being administered? Your thought?
No, no. So that there are several reports that Omicron is actually not quite protected by the vaccine, but
Right.
it is also true. Omicron or Delta, the vaccinated individuals are the sign is severity is much lesser.
Even in Omicron. Even in Omicron.
Even in Omicron, that is
Right.
expected. But you have to always keep in mind that comorbid situation, the age factor, and the individual immunity of that particular person. So unless the hospital network gives us that information, it is too early to call.
Hardly to say. Final question. Many feel that Omicron is the Santa this New Year, this Christmas has brought. Some feel is the Satan, the Santa or Satan phenomena. What's your take on that? Is it the vaccine we were all looking for, the Omicron?
No, no. So as a scientist, what I will say that I can see now more and more people are getting vaccinated.
Right.
More and more people are already infected. So in any kind of an epidemic, if you just go by to a Spanish flu, so at some point they will have this resistance power in a major population. So virus will definitely run out of host, and that's the time we are actually. So it's possible since Omicron is actually infecting more people,
Right.
we might achieve the herd immunity much
Right.
faster compared to whatever. And at the same time, the vaccinations is also going on in a rapid speed. So at some point, we will probably reach a point where the virus will run out of the host.
What's that point? That's a big question. Your take
That's a
on that?
big question. That's a big question. And I
Whole world
also
is whole
have
world is trying to find that answer. What is that time when virus will finally run out of its stream?
yeah, I mean, we were actually, I was also reading in some places that maybe middle of this year we'll have a much better situation.
Your voice lost in between. You were saying that.
No, I said I was also reading somewhere that maybe in the middle of this year, the situation will be much better.
Okay, so it means that around middle of year means not far away, few months, but with IHU and other things are emerging, and as you said, that so many sublineages of Omicron
But I
coming
think
up. Yes, please.
one thing I think you should just say it that no matter what variant comes, if we wear mask, if
Yes,
we maintain social distancing, if we avoid crowd, I think we are safe. So I think it is in our hand.
but then the life will change. Uh, Wearing
Life
mask is
has
it
to be
okay?
reprogrammed. Life won't change. We'll
Life
just
will be programmed
reprogram the life and we enjoy life, but in a different way.
in a different way. Finally, not really any question, but Dr. Das, so many people are hearing you. To the common people, the public health experts, doctors, your comment on the situation of Omicron and how to take it from there. Your suggestion, your recommendation to the kind of stakeholders at large.
That's basically, I would again reiterate that, there is no reason to panic, but you have to be alert. One has to be careful and one have to avoid an unwanted situation. So, and that is in our hand. COVID appropriate behavior, we should go back, reprogram your life, reprogram. So the life cannot really stall. So it has to go on, but at the same time, you reprogram it, you reroute it so that we can actually defeat the virus.
Finally, another, as you said, because a lot of people from other countries will be hearing you as well, that you have any connections with the other South Asian countries, how things are happening out there? INSACOG has a linkage with the
We,
South
we,
Asian level?
we are definitely, we routinely meet with this international consortiums and we are in linkage with them. But
Right.
in addition, we are actually contemplating to have one consortium extended to the neighboring countries
Okay.
in Southeast Asia and others, so that we can have a better network and we can also help them if they are not
Right.
detecting the virus at this time. And we really extend our expertise to those countries and help
Bangladesh
them develop.
expert just told me that they have just 10 institutes for doing genomic analysis in Bangladesh. Entire of Bangladesh, only 10. So definitely your help will be extremely welcome out there.
Definitely, if we can just get any request, it helps us to follow up with that request.
But it has to be, I think, a government level
Definitely,
decision.
it has to be from the government level. So if any countries wants to do that, then you will have a regional consortium for this.
But you are already in touch with the experts and genomic analysis around the,
That we already, I mean, we already have routine discussions with the
and the
UK, with the
trains are
US,
similar,
and the
Dr. Dash,
European
according to
Union.
your
Yes.
input. The trains are
Huh?
similar. The
Yeah,
trains are
yeah. I
similar.
mean, yeah. I mean, definitely we discuss with them, and then we are in somehow consultations with them.
Okay. Thank you, Dr. Dash, for your time. And it's been wonderful speaking to you. And you have mentioned so many points. And the most important point that you have mentioned, when we are in media so agog with the figure, it's 70%, 80%, but quality of sampling is extremely important. You need to ensure that each and every part of the country is represented in the sample, simply because the virus that is still not known, the enemy that is still not known, can be identified in time and can be countered. That's, I think, the very important lesson learned from this today's discussion. But I think also there are a few pointers. I think definitely you will take them on. What the public health experts are saying, that if the data of INSACOG can be shared more frequently, or more, I think I would say accessed in a better manner to the stakeholders at large, then they'll be understanding what's happening. And as you already said, that you are being in process of building up a network with the physicians. So that is also, I think, a very important development. And third is that you are also thinking about forging a beyond national level, a regional level kind of network. That's government has to take a call on that. Uh, I think extremely important point. We will keep on talking. We kept on talking. We'll keep on talking. Thank you for your time, Sudas. We wind up here. Thank you. Thank you.
Thank you. Namaskar.